Hello students, I am Varsha Mistry, Assistant Professor in Microbiology from KK Senior College, Nasik. In this lecture, we are going to study about the acid-fast staining. Acid-fast staining is a type of a differential staining which differentiates between acid-fast and non-acid-fast microorganisms. Now, why there is a necessity to differentiate between acid-fast and non-acid-fast? Acid-fast are those which do not get decolorized by acid treatment, and non-acid fast they get easily decolorized by the acid treatment. And it is important to visualize the mycobacteria under the microscope. Now these mycobacteria, these are mainly the pathogenic microorganisms responsible for tuberculosis or the leprosy. So it becomes necessary to visualize these microorganisms in a stained preparation, as these are difficult to stain by simple staining or by gram staining. So it becomes necessary to apply a type of a stain that is called as the acid fast staining. So it is a differential staining and it was first developed by Ziel and it was then modified by Nielsen. So this is also called as the Ziel-Nielsen technique. Okay. Or it is also called as the Ziel-Nielsen carbol friction staining, that is ZNCF. Nielsen in 1883 used Ziel's carbol friction and heat and then decolorized with an acid alcohol and then counterstained with methylene blue. Okay, and thus the Ziel-Nielsen staining that technique was developed. Okay, so as I told you, it is a staining to differentiate bacteria into acid fast and non-acid fast groups. This method is used for those microorganisms which are not stained by the simple or gram staining, particularly the member of genus Mycobacterium which are resistant and can only be visualized by acid fast staining. Now what is the principle of the acid fast staining? Now when in a acid fast staining, first of all the smear, okay, again the regular procedure of our staining procedure, take a clean and crispy glass slide. Prepare a smear under the aseptic condition, okay, air dry and heat fix. Then the smear, it is stained with the carbol friction. Okay, it is the stain which is used. This carbol friction means it is this acid friction which is prepared in phenol, and phenol it is called as the carbolic acid. So that's why the stain is called as the carbol friction. It solubilizes the lipoidal material present in the mycobacterial cell wall, but by the application of heat, the carbol friction further penetrates through lipoidal wall and enters into the cytoplasm. Okay. As this mycobacteria, they are having a thick, or no, we cannot say thick, but a tough cell wall which is composed of the mycolic acid. It is like a wax-like compound, and so as the cell wall is tough, the stain cannot easily penetrate. Okay. So that's why in this staining we apply the accentuators. Accentuator is one of, that is the heat, and the another one is the carbolic acid, that is the phenol. Okay. So when we apply the carbol friction plus heat, what happens? It solubilizes or it softens the lipoidal material, that is the mycolic acid in a mycobacterial cell wall, and by the application of heat, then the carbol friction it penetrates the cell wall and it enters into the cytoplasm. After that, all the cell appears red in color. Then the next, the smear is decolorized with decolorizing agent, that is the 3% HCl in 95% alcohol, or even we can use the 20% sulfuric acid. But the acid fast cells are resistant due to the presence of the large amount of lipoidal material in the cell wall, and thus it prevents the penetration of decolorizing solution. Okay, so when these acid fast microorganisms they get stained, they get permanently stained. And the non-acid fast organisms, as they lack the lipoidal material in their cell wall, due to which they get easily decolorized and leaving the cells colorless. Then these decolorized cells, to observe them, again the smear is stained with the counter stain, that is either the methylene blue or even we can use the malachite green, and only the decolorized cells they absorb the counter stain and take its color and then appears blue, while acid fast cells retain the red color. Now what is the procedure of the acid fast staining? First of all, The prepare smear on a clean and grease-free slide. As I told you, allow the smear to air dry and heat fix. Then apply the alcohol fixation. Okay, sometimes means this heat fixation, or even we can use the chemical agents as the fixative agents. Okay, so the alcohol fixation also recommended when the smear has not been prepared from the sodium hypochlorite treated sputum, and which will not be stained immediately. Mycobacterium tuberculosis is killed by bleach, because in this acid fast staining, when you are taking the sputum sample, that sputum sample, it contains the highly pathogenic. If it contains the highly pathogenic mycobacteria, then it should be killed. It should get killed. Otherwise, it may harm that person which is performing this procedure. Okay, so this mycobacteria is killed by bleaching during the staining process. And heat fixation of untreated sputum will not kill the Mycobacterium tuberculosis, but whereas the alcohol fixation is bactericidal. Okay, so this alcohol fixation is also preferred. Then cover the smear with a carbofuction stain. Heat the stain until the vapor just begins to rise. As I told you, heat that is used as the accentuator. Do not overheat. Allow the heated stain to remain on the slide for the 5 minutes. Heating the stain. The great care must be taken when heating the carboflux stain, especially if staining is carried out over a tray or other container in which highly flammable chemicals have collected from the previous staining. Only small flame should be applied under the slides using an ignited swab previously damped with a few drops of acid or alcohol. Acid alcohol or 70% volume by volume ethanol or methanol. Do not use a large ethanol soaked swabs because this is a fire risk. Then wash off the stain with the clean water. Then cover the smear with 3% volume by volume acid alcohol for 5 minutes till the smear is sufficiently decolorized, that is the pale in color. Okay. Then wash it with the clean water. Cover the smear with the malachite green or the methylene blue, that is the counter stain, for 1 or 2 minutes using the longer time when the smear is thin. Wash off the stain with the clean water. Wipe the back of the slide and place it in a draining track for the smear to get air dry. Examine the smear microscopically using the 100x, that is oil immersion objective. So what you will observe? You will observe red colored acid fast bacteria and the background, which is blue colored. Means the background we observe that is a blue color because the sputum, it contains the various particles along with the epithelial cells of the respiratory tract, वगैरह. Okay, so this background appears blue in color, or the normal flora of the respiratory tract is also present, so that appears blue in color, and the acid fast bacteria, they appear red in color. Okay, so the examples of the acid fast are Mycobacterium tuberculosis, Mycobacterium pneumatis. Non-mycobacterial bacteria are Nocardia, then Cryptosporidium. These are the examples of the non-acid fast bacteria. Okay, so this is about the acid fast staining.